I'll be talking about something that's very close to my heart, which is human evolution. And we're talking about both the fossil evidence for human evolution, something that's become of increasing value and accessibility in paleontological studies, is the use of DNA in paleontology. So I'd like to begin by talking about birds. And the thing is, I'm a bird watcher, and it's very easy to be a bird watcher in Ohio this time of year simply because this is when all the warblers are coming through. And I think the idea that I like about bird watching is that there are so many different species that you can see. Even on a bad birding day, just walking down your driveway, you'll actually see a half a dozen species. And if you take your binoculars out and you listen carefully, it's easy to see hear 12 species or see 12 species within a very short period of time. And the other interesting thing about the birds is not they're colorful and they fly and they do all sorts of things that I would like to do. I'd love to be able to fly. But they also have a tremendous diversity of adaptations. You'll see some of them are eating seeds. Some of them are eating seeds on the ground, some up in the trees. Some of them are eating insects on the ground, or the woodpeckers are trying to pull them out of the bark or in the wood of the trees. And you'll see the carrion feeders or hawks circling around. And so there's just a tremendous amount of diversity. And what we find is, in order to document this diversity, we get our Peterson's Guide. And I just checked my Peterson's Guide I bought in 1984. And it is very worn out. I've already gone through 2 layers of tape on the spine. And so here is the birds. And so it documents this variation of all the species that we see. But now imagine that you're a bird and you want to go human watching. And so that it's a very, very different story, but what you do is you go out and you get your Peterson's Guide to Worldwide Human Documentation. And what you're going to find is this is a very thin book. And the reason it's a very thin book is if we were to create a web page or a page of this and look for human diversity and do a thumbnail sketch of what humans are, we look at the number of speciation we have here. So species diversity for the species Homo sapiens, just 1 worldwide. And if we look at the conservation status, I think we're very abundant. And some of the other characterizations that we have here is that we occupy a tremendous diversity of habitats, from the tropics to the poles. Low altitude, high altitude, we have a diversity of diet. We eat pretty much anything that's in front of us. We're very social animals. We live in social groups that range from family groups to actually cities where millions of people living in a small or confined area. We are generally non-migratory, except when we get in the 7th decade, and then I'm going to migrate back and forth to Florida. But then we are chronic and acute tool users and makers. It's part of our adaptation, is manipulating the world by the use and manufacture of tools. And using the phraseology of Peterson, it's our only bipedal mammal. And so it's, again, some other characteristics we can see here, the stature and diet. And what I'd like to do then is describe some of the characteristics of humans. So has there always been just one species of human alive at one time? It's very difficult to me to imagine that there is another species of hominid that could be existing today, because our adaptation is so pervasive. We live everywhere. We eat everything. We occupy every niche. We don't partition down our niches like the birds do, that it'd be difficult to see another human species that does what we do. Now the issue is when did this uniqueness occur? So what is it about humans that make us so unique? And we're going to be discussing some of these changes that have been occurring in the past 2.5 to 3 million years. Other things is, when did these adaptations arise? So we'd like to look at the sequence of changes, and that's what paleontology and archaeology can help sort out. So the traditional analyses of trying to reconstruct the behavior and biology of our human ancestors is based on paleontology looking at the bones, or archaeology looking at the cultural remains of our ancestors. And this has truly been the traditional approach, and I'm a paleontologist, and I used to be an archaeologist. And so what we find is that this is the way that I try to figure out the past. However, what we find is that with recent advances in our understanding and sampling and analyses of DNA, we can now understand not only understand better the aspects of adaptation and variation and phylogeny of modern humans, but with our capability of sampling ancient DNA, that we can now extend these studies back into the past, going back now to almost 500,000 years ago. So what makes us human? We'll describe some of the characteristics that are familiar with us and in some broad sequence. Break it down to the early adaptations. and this is walking on two legs and having a small canine tooth. And this says something about our locomotion, says something about our reproductive behavior, believe it or not, says something about what niche we're occupying. And this was the focus of Dr. Haile Selassie's talk last week. The middle adaptations were between about 2.8 million years ago and a couple 100,000 years ago, are the adaptations we'll be talking about tonight. And this includes the manufacture and use of stone tools, which arose about 2.6 million years ago. The diet regularly included animal remains. We became carnivores, and again, this also occurred about 2.6 million years ago. We're the only primates that regularly eat meat, although chimpanzees will occasionally do so. Our brain size are much larger, and this began taking off about 2 million years ago. And then we were able to occupy habitats beyond the tropics. We moved out of Africa about 1.8 million years ago and spread throughout much of the old world. Then our recent adaptations, or those adaptations that are associated with Homo sapiens, our species, include that we left Africa. Our species, Homo sapiens, left Africa about 60,000 years ago. We began to settle down in, some populations moved into large, dense settlements. This created changes for political structure and social roles in humans, much more complex than in other animals. And then we also get into things like plant and animal domestication, both dogs for companionship and whatever else we need dogs for, but also for a variety of plants and animals for food resources. So just as a quick review of some of these changes, it's a timeline of human evolution. We'll begin the earliest stone tools, which are known as Oldowan tools at 2.6 million, the origin of carnivory. Earliest Homo habilis, or the origins of the first named species of the genus Homo, the genus in which we are classified. Then about 1.8 million years ago, the earliest Homo erectus, again out of Africa, and the development of Acheulean stone tool technology. Then about 1 million years ago, or perhaps a little older, humans, or our ancestors, moved into Europe. Then we get the origins of a species of human ancestor referred to as Homo Heidelbergensis. This is an unfamiliar name for a very familiar fossil. It used to be called archaic Homo sapiens. And then we get to Neanderthals, which is our first foray into paleontology and human evolution as a child, those brutes that lived in Europe. And then we get the earliest Homo sapiens, truly our species, Homo sapiens, about 200,000 years ago. And then Homo sapiens themselves migrated out of Africa about 60,000 years ago. Now, so I'd like to just document some of the anatomical changes that have gone on in human evolution. And one of the things that we can see is that there's been a marked change in brain size. Beginning about 2.5 million years ago, although documented in the fossil record about 2 million years ago, our brains increased incredibly. Now, Lucy's brain size is about 375 cc, so we can see that Lucy would be down about here. And so these are the Australopithecine species down through here, and our brain size are about 400 to 500 cc's. Our current brain size is on average about 1350 to 1400 cc's. So then beginning about 2 million years ago, we can see that there's a steady increase in our cranial capacity. Each one of these dots is a fossil hominid cranium. And the other thing that describes our adaptation is the use of tools. Now, our tool set is much more complex today than it has been at any time in the past. So in the lower left corner, we have a Swiss Army knife, a tool that many of you have in your pocket right now. And we can see that this has a greater diversity of tool types than probably were had by human ancestors until several tens of thousands of years ago. Pliers, knives, scissors, these were things that were unknown in the fossil record. But then we get to some other, we've been able to elaborate on our tool technology. And so we now have television so we can communicate long distance with individuals. We have cars so that we can carry ourselves and our belongings back and forth to different areas. We can even envision and create rockets so that we can leave Earth, something no other species can even conceive of or consider doing. And one of the important adaptations that we find here is the stove or fire. Fire is an ancient adaptation, but what this does is this allows us to occupy a variety of new niches because it allows us to eat broaden our diets. What we do is we can detoxify food So we can eat things that we previously couldn't. We can break down their consistency of food so that we can eat things like potatoes and yams. And then we can also kill the bacteria that grow in the food. So that what we find is fire is a very central adaptation to humans. And it just seems like a simple thing to us now, but it truly was a breakthrough adaptation. So some of the other adaptation we'd like to look at, so look at the earliest tools. This, in fact, is one of the earliest stone tools that has ever been recovered. This is from a place called Gona in Ethiopia, where I've been working since 1999. This is referred to as an Oldowan stone chopper. It's what they do is they took a cobble from the river, took another rock and broke the cobble, and it created a sharp edge on one edge of the stone, and it was smooth on the other we think, for holding. And this was the pinnacle of technology at 2.6 million years ago. And this truly was a breakthrough. The Gona stone tools were recovered from this area. This is actually the site, or the region, where the Gona tools were located, down where the people in the lower right-hand corner are situated. And so what we find is these individuals would collect the stones in the riverbed, carry them to the riverbank, and then break them apart, and then use this for the various things that the early Homs used them for. And so that all these archaeological locations where we find these early stone tools in the Old One are along these riverbeds, which is where the source of stones came from. And so this is what they're using the stone tools for. This is an antelope shin bone, a tibia. And what we find you can see in this image, his shin bone is 2.5 million years old, as part of the team that excavated this. And so we find that this is a very battered bone. And you can see, if you look carefully, there are a series of scratches here. These scratches are actually cut marks that were made by a stone tool as they were cutting the soft tissue off the bone. Another thing we can see is these big areas right here, this one and another one here. These are percussion marks, where they were taking a stone tool, a cobble, and breaking open the tibia, trying to get inside, to get the marrow inside. And we can see this edge of the tibia here, the distal end, is actually the green break, and it was broken open by these early hominids so that they could actually get the marrow out of this. So what we find here is that 2.5 million years ago, they were developing a new technology, stone tools, and they were occupying a new niche, getting out there and exploiting animal resources. So the question becomes, who was this early stone tool maker? So back in the late 50s and through the early 60s, the team, Lewis and Mary Leakey, working in Olduvai Gorge in northern Tanzania, discovered a series of fossils, and this is one of the reconstructed specimens, and called by the great name Olduvai hominid 24, or Twiggy. And we can see here that this is the map located in upper Tanzania. And so that the Leakeys found a series of these early primitive hominins in association with those Oldowan stone tools. And that's why the Oldowan stone tools are called Oldowan, because it's named after Olduvai Gorge. And we find that there are these early hominids are actually located throughout the East Africa and down into South Africa. And there are not very many, but they existed for a period of time and had a very characteristic distribution and stone tool technology. And so this means we've got to create another page for our guidebook to humans and our ancestors. So we name this human ancestor Homo habilis, or handyman, because it could use its hands for making stone tools. Family Hominidae, the earliest good evidence we have is about 2.3 million years ago, and it went extinct about 1.6 million years ago. It's an omnivore, but its brain is quite small, still 500 to 600 cc's, well below the human average. But it is much enlarged over the Australopithecines, and they have adopted carnivory into their diet.
What do they use the marrow for?
Eating food. It's filled with proteins and fat.
They would crack the bone?
Yes, they would. And so that's why they'd use these cobbles in order to shatter the bone so that they could break it open and get into the delicious marrow inside.
On the previous slide, did I see that they weighed like 50 to 100 pounds or something like that? I'm trying to get an idea of like how big they were, and I know you said their brain was about 1/3 the size of ours, but can you otherwise describe them?
Okay, so what they did was so the question is, what did the early Homo habilis look like? We do not have, surprisingly enough, we don't have very much of their postcranial information, postcrania, their bones. We have one partial skeleton, which is illustrated here on the lower left, OH62. This is from a very small individual. How it would differ from Lucy would be that this individual is probably the same height, maybe a little taller than Lucy, so that means it's about 3 feet 9 inches, maybe 4 feet tall. Relative to Lucy, her legs were longer than Lucy. Lucy had short, stubby legs. Homo, after about 2.5 million years ago, there was an elongation of our lower limb, perhaps as a refinement of our bipedal locomotion. OH62's arms are also quite long. So whereas our arms stop here and end about mid-thigh, theirs probably went a little bit lower, a little lower on the thigh. And that's probably just a remnant of their early Australopithecine adaptation. It's difficult to say much more about this simply because we don't have that much unambiguous information about Homo habilis. We can make some inferences about how big the males and female differences are based on the differences in size of the crania, but still it's an imperfect representation. So what we're doing is we're using what little information we have, the size of the femur, the size of these limb bones, also the size of the teeth, and then we're comparing that with living animals and seeing what the body size of extinct animal is using guidelines based on living animals.
Bringing down a wild animal for someone 3 feet tall, did they throw stones at them?
Well, the big question is, are they hunting or are they scavenging at the time? And it's a debate that truly goes back and forth. You're right, because if you can imagine is that a small biped is a very slow animal, and it would have difficulty tracking down or chasing down other animals, unless, of course, it focused on eating smaller animals. Turtles are a very interesting food source for these animals. What we find is that after about 3 million years ago, after humans adopted carnivory, the size of tortoises go down. Before 3 million years ago, we'll find these giant tortoises that are over 1 meter in diameter. After 3 million years ago, the frequency of those tortoises is much less. So they're probably getting the easiest accessible food first. Another thing you could do is go out and get scavenging. After a kill comes down, there's a kill. You can imagine the hominids can get together, they're social animals, and that they can perhaps chase off the larger predators. We see this occurring in Africa when things like jackals can chase off lions. And if they are throwing stones, which is just conjecture at this point, then you can feel that they've got an additional tool in order to be able to chase away the animals at a kill. The problems you face, though, is that at a kill site, it's everybody on the savanna, if you're out in the grasslands, knows when the kill site's occurring. First animals that identify it are the vultures. The carnivores also recognize where the vultures are, and also with smell can follow down to the kill site. So what you'll find is that a scavenging site is actually an arena where you'll have multiple different types of carnivores living at one spot, meaning that it's a very dangerous space. So you might want to go in and snatch out a piece of the meat from the kill, but what that means is another animal may want to snatch you up too. And so you get a twofer, the scavenged animal and then the fresh hominid.
Is there any evidence they were using clubs rather than just throwing a stone?
Is there any evidence they're using clubs? What we find is that the answer is no. There's no evidence, but that doesn't mean that they weren't using something like that. Simply because that these the location of these sites are they're open air sites, and organic materials just don't preserve very well for a long period of time. Okay, what I'd like to do is then move into the next section, and we're going to begin again with tools. So we're looking at human evolution now of tools that arose about 1.8 million or 1.75 million years ago, and this is the Acheulean technology. And the Acheulean tools are characterized by two main tool types, and that's the hand axe in the upper right, and then we also have what's referred to as the trihedral pick. The Acheulean hand axe is a very long-lived tool, again, first found about 1.75 million years ago, but it continues through up until about, 200,000 years ago or so, variously in Europe and in Africa. And this is a tool that is usually either took a long cobble or a flake of rock, and they flaked it on both sides so that it was a biconvex cross section, and it had a blade or a flaked edge going around here, forming something of a knife, and it often had a more pointy edge and a more rounded edges. This is a very derived Acheulean hand axe from about 7.78 million years ago, but the early ones are truly primitive. The other tool is this trihedral pick, which instead of having that biconvex cross-section, actually has a triangular cross-section. And so again, it's got a point at one end, it's got a blunt edge at the other, and so that these are very common types of tools. So what they've done is they've added to their toolkit, we now have a better quality knife, and now we get something that is perhaps a poker, a digger, or some type of point for breaking into bones or accessing food in another way. So the toolmaker, the earliest toolmaker of Acheulean tools is Homo erectus. Now Homo erectus was discovered in 1891 by Eugene Dubois, and it was the first truly primitive human ancestor that was discovered and recognized by scientists. Dubois was a Dutch physician. He lived in the Netherlands. But he had read Darwin, and he was interested in human evolution, and he thought that he would like to go find the fossil evidence of human evolution, and he thought that these early ancestors lived in a forest, and he thought that the orang might be a good model for human ancestors, so he decided to go where he thought this would be, and he went to first Sumatra, and then he went to Java for the express purpose of looking for fossils. In fact, he got reassigned to this area on purpose. And after a couple of years of surveying for fossils, he identified a locality where ancient animal bones of extinct animals were eroding out of the bank of a river. He hired a team, and they began excavating and looking for these fossils. And soon enough afterwards, they found a femur, which is a thigh bone, which is very similar to us. This is a pathology on this individual. He found some teeth, and he found the skull cap. Now, the femur was recognizably human. The teeth were very similar to human, but the skull cap represented a brain or a head that had a much smaller brain than did modern humans. But he recognized it was a human ancestor, and he named it as Pithecanthropus erectus. Pithecanthropus means ape man, and erectus means walking, erect walking ape man. And he was absolutely right. But nowadays we call it Homo erectus instead of Pithecanthropus erectus. And Homo erectus is now recognized throughout a variety of different localities in the world. We have in Java, down in southern Africa, Tanzania, Ethiopia, Kenya, West Africa, Turkey, Dmanisi, this up here in Dmanisi in the Republic of Georgia, and then we get a series of sites in China, including the Peking Man up by the cave sites in Beijing. Now what we want to do is, because this is a field guide, we want you to be able to identify your Homo erectus when you find one in the field. Its brain size is much smaller than humans, with quite a big range from 600 to 1200 cc's, about 85% the size of modern humans. The vault shape is different. It's a long, low vault, and that reflects its brain size and also the geometry of the way the vault and the face are hefted together. It has brow ridges, which are ridges of bones atop the eyes. The other associated with this is behind you have a very flat forehead or a sloping forehead. There's oftentimes a sulcus or a groove behind the brow ridges and separated from the forehead. And then we have cresting and ridges, which are characteristic and diagnostic of Homo erectus. First, on the top of the vault, which is shallow, also some transverse cresting. But we also have some thick cresting here in the back of the head where the neck muscles attach onto the back of the skull. And finally, the widest part of the Homo erectus cranium is just above the ears, whereas if you look at the widest part of our vault, it's up high in the parietal regions. So now you know the field signs for this. Now, a number of researchers have characterized Homo erectus as being human-like from the neck down. And I think that that's not an accurate representation. In fact, Homo erectus was not modern human from the neck down. It was in a transitional form, a long-lasting transitional form between the early Homo habilis and modern humans. And one of the examples we can see of this transitional form is from a pelvis from the gonocyte. And so this is Lucy here. This is a modern human here. And then we have this Homo erectus pelvis from 1.2 million years ago. And one of the things I'll bring your eye to is the shape of the birth canal. And we can see in humans, this is a human female, we've got a large birth canal, and the reason we have this is because we birth large-brained offspring. Well, Homo erectus, their brains were much larger than the Australopithecines, so they too had these modify the obstetric pelvis to allow these big-brained offspring to get through here. So we can see that there is some transitional here and that there is some adaptation going on both in the pelvis and the head, as well as the behavior of Homo erectus. So let's add another page to the field guide. Again, Homo erectus, conservation status, unfortunately extinct. Originated about 1.8 million years ago, but its last appearance was in Java about 140,000 years ago. Again, it was an omnivore. Brain size was quite small, 550. It's up almost 90% the size of ours. They, again, lived in Africa and well into Asia. And Homo erectus was the first species that really got a chance to move out of the tropical environment. This was the first species that left Africa. So it was a very important adaptation. There's no evidence of sedentary occupation, and question mark fire, we don't know, and they're associated with the Acheulean tools. Now, Homo heidelbergensis is, again, this is the hominin you know, but you didn't know you knew it. And we've seen these fossils because they're very well, a couple of them are very well preserved, and they look like, I don't know, Homo erectus on steroids. But they're distinguishable from Homo erectus because they have a larger brain, and they've got a different face. Their face is hafted to the skull a little differently. There's some fine details in the morphology of the face, but it's a slightly more, it can be a slightly more gracile face than we see in Homo erectus. But now we want to distinguish it from Homo sapiens because it does have a smaller brain than modern humans. The face is characteristically different. It still projects in front of the vault a little bit, whereas the human face is underneath the cranium, and it is more robust. And we can also look at the vault. It's longer and lower than we see in modern humans. And so these are poorly represented, with one exception, throughout Africa, Europe, and China. Some call the same species Homo rhodesiensis in Africa, but that's neither here nor there. But it begins about 600,000 years ago, probably a little older, and it goes extinct about 200,000 years ago. And it seems to be a reasonably good morphological intermediary between Homo erectus and Homo sapiens. And Homo heidelbergensis, again, we find this in Europe, including up in England, but France, Spain, Germany, Italy, Greece, a couple places in Africa, and then we also find some in Asia. So it too is a widely distributed human ancestor. Now, one of the sites I'd like to talk about for describing Homo heidelbergensis is at a place called Atapuerca, and it's the Cima de los Huesos. And this is the pits of bones in the Atapuerca Mountains in northern Spain. It's dated at, some dating put it at greater than 530,000 years ago. Some other dating puts it older than 300,000, so there's some discussion about that, although I tend to support the older date. And the Cima is in a cave, and the cave, the bone deposits, the pit of bones, was discovered by spelunkers who were looking for cave bear fossils. So they crawled in a cave that was half a kilometer long, got to one end of the cave, and they found a pit that was many meters, or several meters deep. So they climbed down into the pit and they started finding cave bear bones. So they did a preliminary excavation, discovered a hominin mandible, brought this back to researchers, and the researchers have gone back down and excavated in the pit, and we now have over 3,500 hominid bones from this single area, representing over 28 individuals. And you can see that the preservation is pretty good. So we have males, we have females, we have children. And these individuals, they probably did not fall into this pit. It's most likely that this was a mortuary practice, and this is where the individuals were being, I don't want to say disposed of, but where they were placed after death. The other curious thing is that there are over 150 cave bear fossils, individuals, also in this pit. And so the question is, well, why? Well, cave bears are carnivores. They're opportunistic carnivores, and so if they smell rotting flesh, they'll walk down this cave, get to the edge of the pit, and fall in. And they're trapped. And so that they find evidence of the cave bears trying to scratch their way out of here. And so another cave bear had fallen, another cave bear had fallen, so we have 150 cave bears. There are also additional carnivores fossils in this area, in this site. Another interesting fossil we want to talk about is Boto. Boto is from Ethiopia in the Middle Awash Project area, dated about 600,000 years ago. This individual has a brain size of about 1,100 cc's, and this has the largest face of any human ancestor. But the curious thing about Boto is if you look very carefully, in this region highlighted here, this is its left cheekbone, And if we look right through here, we'll see some scratches. And so that these scratches are in fact cut marks. Some individual 600,000 years ago took a sharp stone tool and began slicing the flesh off of Boto. Now, whether this is mortuary behavior or whether it's food preparation behavior, we don't know. But it's very interesting that we see this type of cut marks not only on prey animals and food animals, but also on conspecifics. So then we add another page to our field guide, Homo heidelbergensis, and we can read through the anatomy, perhaps ancestral to Neanderthal, Schulean tools. And again, primarily, we know a lot of this individual from around the SEMA. The Paleontology, now we're getting to Neanderthals. These are familiar human ancestors, or human cousins rather. Well, kissing cousins perhaps. And so this specimen right here is the old man from La Chapelle-aux-Saints. We know he's old because we look at his teeth. Maybe his post-canine teeth have worn out. They've fallen. He's lost some early in life. All the bone has been resorbed away. We can see incredible anterior tooth wear. And this is a composite skeleton based on a number of Neanderthal specimens that have been recovered. The distribution for Neanderthals is in the shaded area, and we find them again throughout Europe and getting into westernmost Asia, getting down also into the Near East. And this is the distribution based on known fossils of Homo neanderthalensis, of Neanderthals. And if we do our field guide for Neanderthals, what we do is, first thing we do is look at brain size, and their brain size is in fact larger than modern humans. Our brain size is about 1350 to 1450. Their average brain size is 1600 cc's. We notice that they still retain the brow ridges. They have a longer and lower vault than do humans, although if we look at it from posterior view, it's quite round. It's a globular vault. And there's some other facts. They don't really have a chin in here, but there's other identifying characteristics we can use, but we would never mistake a Neanderthal for a human, or so we say. Now, Neanderthals are, again, we have evidence that they lived in caves, and they brought their prey into caves, and that's where they prepared them. And so at a cave called Moulin-Gercy in France, the archaeologists were excavating the bones. They found lots and lots of bones from bison and deer, and they noticed the breakage patterns of the bone and the cut marks and the way in which the food was processed, the animals were processed for food. But they also, in the course of excavation, they discovered a number of human bones. These are fragments from a human femur. These are human cranial bones. This is a mandible. This is a humerus, or an upper arm bone. And we find scratches made by sharp stone tools, scrapings on here. We can identify what muscles were cut by where the scratches are. This is the inside of a mandible. So what they were doing was they're cutting the muscles out that were supporting the root of the tongue. Right here, they were cutting the muscles that surround the elbow. And so what we find by analyzing the non-human bones and the Neanderthal bones is that they were being processed in the same way, and they're being processed for food. And it's very interesting. So this is some of the best evidence we have for cannibalism in Neanderthals. And so since we've gone back and looked at other sites, we find that in fact there are a handful of other Neanderthal sites that also exhibit cannibalism. So we add our other page, the Neanderthals. We notice the big brain. They're also very tall, very robust types of animals found throughout Europe and Western Asia. Now I'd like to talk about modern humans. Now we're going to get to modern Homo sapiens. That's us. One of the fossils we're going to talk about is at Herto in Ethiopia. This is the Bori Peninsula. This is where Dr. Haile Selassie and I have worked. One of the fossils that we recovered at the site is this cranium here. There's one of a handful at an open-air site. And we call it Homo sapiens idalto. Idalto is the local name for elder. So this is the elder human, dated about 165,000 years ago. Although the earliest evidence of modern Homo sapiens is in southern Ethiopia at 195,000 years ago. And so the question is, well, why are we calling this a human? And there are a series of criteria that we can examine: the brain size, the shape, the globular vault of the neurocranium. We can look at the forehead. We can also identify the facial shape and some details of the face, something called a canine fossa. And these are signatures of modern humans. But it's not exactly like modern humans, because what we do is we do have the larger brow ridges. the overall cranium is more robust, and the tools are not the same as we see in much later Homo sapiens. But this is a recognizably human individual. This is a reconstruction by the artist J. Maternus, and you can see that he looked at the anatomy, anatomic relations on here, and drew a reconstruction. And you can see that it becomes a very, very familiar type of individual. Now, before the break, I'd like to talk about one more very, very interesting member of our lineage, or not our lineage, but of our family tree. And this is Homo floresiensis, named because it was found on the island of Flores down in Indonesia. It's commonly known as the Hobbit. I think it's because it was discovered and published about the same time as the Lord of the Rings trilogy movies were released. And it's from Indonesia, and it's 17,000 years old, or perhaps as young as 12,000 years old, to 74,000 years old or more. And this was found in a cave in Flores. Now, the reason this is a very important fossil for a number of different reasons. One, it lives at the same time as modern humans. So getting back to my question, can there be two types of humans living someplace on Earth at one time? And the answer is, well, yes. But the reason we got yes is Flores is on the east side of what's known as the Wallace Line. And so even though it seems it's very close to Java, in fact, there's a trench that runs between Flores and the islands to the west. And what this means is even when the water levels drop during peak glaciation, there's always a channel here. So there's never a land bridge between Java and Flores and points east. So this means that the animals and many of the plants that are found east of the Wallace Line are different from those that are found on the west. So that really is a biogeographical boundary. The other curious things about the Hobbit is, well, as you know, hobbits are small with big feet. This guy is small with big feet. It's probably only about a meter tall, and we know this from several individuals. It has big feet for its body size. The other thing we find is that its brain size is about the same size as Lucy. So whereas our brain size is about 1350 or to 1400 cc's, its brain size is about 380 or 400 cc's for this individual thing. We also find that it's associated perhaps with stone tool technology because there are stone tools there. So it's very difficult for us to envision how such a small-brained animal can make such complex stone tools. The other thing is, what's its ancestor? Is it a descendant of Homo erectus? We don't know. We'll have to learn more from this. Unfortunately, the specimen, even though it's young, it's 17,000, we cannot get DNA from this specimen. So the question is this a small-brained individual, or is this the village idiot? We just don't know. We've got to go out and find a couple more individuals, a couple more crania. We have to add our page to the field guide for humans. Really not much we can add. And just want to review before the questions is just the species we've looked at: Homo habilis, Homo erectus, which you've talked about, Homo heidelbergensis, Neanderthals, humans, and then floresiensis. I want to emphasize this is a thumbnail sketch of what's going on, and I'm doing great disservice to the complexity of fossils that are actually recovered here and the nomenclature that we have here. The understanding is much more complex than illustrated here.
I noticed in most of the skulls that you showed that there were full dentals. That's really surprising. If you went through this room, I think you'd find a lot of partials or less.
Yes, yes, I know.
How do you account for that?
Speaking from experience, what we find here is that there are a couple reasons why we don't have a full dentary, a full 32 teeth. One of them is because our faces are getting smaller. What we're doing is we're losing our third molars. And so many of us have had our third molars pulled by the dentist because they didn't come in properly. And so that's one of the reasons why many of us don't have our M3s. Another reason why we don't have our third molars is because our face is shrinking. We're also finding, and associated with that, is that people are just not growing their third molars in there. But the other real reason why we lose teeth throughout the course of our life is a couple things. One is as we age, and so that as we get older, we wear down our teeth, and as we wear them down, and then infections can get in the pulp chambers, we wear them down. But the real reason why we have trouble with our dentitions now, why we lose our teeth, is sugar, dietary sugar. And if you look at populations that have a reliance on sugar, prehistoric populations where they have reliance on sugar, like corn, we find these individuals have a tremendous problem with dental caries. Now, one of the specimens that I had to cut out for this talk is there's another specimen of Homo heidelbergensis that's thought to be about 300,000 years old. And you're right, everybody else has a full set of teeth, and there are no caries. But if you look at this specimen from Broken Hill in southern Africa, in fact, it has, just in its upper dentition, it probably has 15 cavities in there. And so that it had some karyogenic diet, some type of dietary sugars in a diet that is very unusual, and we don't see it. But again, we really don't have a problem with our teeth until we start eating sugar.
To go back to the Neanderthals and the cannibalism for a second, is there physical evidence that would support the contention that they were either eating people who were already dead or whacking people in order to eat them?
I don't know. So the idea, are they scavenging or are they hunting their colleagues? We don't know. We don't know. In the individuals that we see here, the remains are so fragmentary, we can't find any evidence of the mortal blow on these individuals. Might be opportunistic. We really don't know. Okay, so what we'd like to do is get into the final section of this. And the final section is actually the changes, the new advances in paleontology. And I'm going to have to plead ignorance on a lot of the DNA material. I am a paleontologist, I'm an anatomist, and I pick stuff up and I look at it. And the DNA materials, the fine details of how these analyses were conducted, how the samples were collected, are sort of beyond my skill set. But what we'd like to do is just report to you so that means ask easy questions. So what we'd like to do is report some of the very exciting research that has gone on. So as I said, morphology is a very useful tool for establishing phylogeny and adaptations, but it really does have its limits. Is that what we find is that the teeth and bones are incapable of picking up fine details of migrations and interbreeding of populations, and because changes in morphology can occur over many, many, many generations, but changes in the genic composition of a population can change very quickly if we have things like in-migration or out-migration or bottlenecks. And the other thing we pick up is that not all populational differences are reflected in the skeleton. It's insensitive to that. And the other thing that confounds this is that when human groups get together, they tend to mate. And so that's why vini, vidi, copulae. But DNA, which has now been able to recover this from fossils, has been very useful for looking at studying the details of human evolution over the past 500,000 years. So scientists have been able to sample DNA from specimens as old as 500,000 years old, and to get enough of the mitochondrial and the nuclear DNA to be able to reconstruct significant portions of sequences, or in Neanderthals, the entire sequence of the nuclear DNA and mitochondrial DNA, so that they can begin to use this as a tool of comparison between different fossil specimens and also between those fossil specimens and modern humans. So this is just as a map of the study of the haplotypes or the different variants in mitochondrial DNA in modern humans. And what we can do is we can examine the patterns of DNA sequences in the mitochondrial DNA, and using this information we can go back and we can predict the sequence of origins of the different haplotypes. And we can string them together so we can understand both the timing and the sequences of changing, so we can document patterns of migration in humans. And if we track everything back, we see human origins again for modern Homo sapiens. The ancestors arose in Africa, moved out into Eurasia, some populations going west, some populations going east, and finally some populations making it into the New World. And then we can also go back and estimate when the timetable was. And based on these data, we expect the first Homo sapiens out of Africa about 55 to 75,000 years ago. So let's go back to the range of Neanderthals, except what I've done is I've modified the title, so this is pre-2007. So this was the distribution I showed earlier. This is a distribution that was revised in 2007 by research by Kraus et al. And there's a cave in Siberia called Okladnikov, and there's very small bone fragments that have come from here, a partial humerus, a couple other elements of the skeleton. That were morphologically not diagnostic of Homo sapiens or Neanderthals. And so they couldn't say anything about its anatomy taxonomically, but they could analyze its DNA, its mitochondrial DNA. And what they found, it shared mitochondrial DNA sequence with other Neanderthals. So now what we did is we made our diagnosis and our allocation, our identification of this small fossil bone based on its DNA. I think that's extraordinary. Then there's another cave which is a little south of of the previous cave called Denisova. It's also in Siberia. And it's been a cave known for many decades. And they did some excavation. Here's a toe bone that was recovered from the Denisovan deposits. And so this time they were able to get not only its mitochondrial DNA sequence, but its nuclear DNA sequence. And they then compared that with other Neanderthals. And not surprising, here's Denisova, and here are a couple of other Neanderthal sites. And they recognized that the nuclear DNA from this little toe bone, again, an anatomically undiagnostic bone, was similar to Neanderthals. So we've identified the full extent of the range of Neanderthals. So I think that's a very interesting topic. And so there's nothing new to come out of genetics. But wait. So let's go back to Denisova. So this is the Neanderthal toe bone, dated about 41,000 years ago. In other deposits in the same cave at about the same time, these both came from around level 11 at Denisova, they found, believe it or not, this is a partial finger bone, and this is a molar. They actually found another molar. They did a DNA analysis of these individuals, and they found it was very different from the Neanderthals. So what we find in one cave, pretty much in the same stratigraphic level, about the same time, is an incredible amount of genetic diversity. And the diversity that people recognized was so great that they thought it might be actually a new species, which we know only genetically and not morphologically. So here's a list of some specimens of which the Denisovan caves specimens were compared against. So this is the Denisovan, that's that little tiny finger bone and tooth. This is that toe phalanx from Neanderthal, and these are the other Neanderthals. If we look at the length of these trees, we see that the toe phalanx, the Neanderthal, clusters with the other Neanderthals. That's very nice. But Denisovan does not. It's quite different, using its autosomal DNA. And all of these are human populations. So we see how the humans cluster together as a separate unit, distinct from that of the Neanderthals. But they're part of a tree, the Denisovans and the Neanderthals, based on the nuclear DNA. But if we look at the mitochondrial DNA, we get a slightly different story. Here's the finger bone and the teeth, and we can see it is more different from the Neanderthals and the toe bone than are humans from the Neanderthals. So the thing is, what we do is, based on the different genetic data, we get slightly different interpretations of the genetic data, depending on whether we're using the mitochondrial DNA or whether we're using the nuclear DNA. But in either case, what we're finding is this Denisova material, known as the Denisovans, are quite different. And so that changes the way in which we look at how individuals distributed themselves across Eurasia. So then we have to create a new page, but unfortunately we don't have a picture, image not available quite yet. But again, it's a new page, and what we have to do now is combine our morphological analyses with our genetic analyses when we're doing paleontology in the Middle and Late Pleistocene. Then things get really weird. All right, so each one of these dots represents a fossil locality that has yielded ancient DNA. Over here, this is the Denisovan cave that we looked at, which is the specimens are about 40,000 years old or so. Then we'd like to compare that with some recent DNA that was collected from the Cima de los Huesos. That's that pit of bones where all the cave bears are. This is 530,000 years old. They got a partial mitochondrial sequence of the DNA from the Cima bones. Now, what's interesting is anatomists have gone out and recognized that the Cima de los Huesos bones are anatomically antecedent to Neanderthals. So you would expect that all of this DNA would be quite similar. What we find is that the DNA, the mitochondrial DNA sequence of the Cima, is more similar to the Denisovans than it is to the Neanderthals. Now, there are some confounding issues again because of the great differences in age, but what it was is we would expect that your natural inclination would be that there would be a similarity with the Neanderthal DNA. So what we find is that in the mitochondrial DNA of that small finger bone is more similar to this much more ancient Cima bone than it is to the toe bone that's found in the same layer. So it's very, very interesting amounts of diversity, and this creates a lot of confusion about how we would explain the biological history that would produce this genetic result. So here's just a mitochondrial tree based on this. This is the SEMA, the stuff from Spain. This is the Denisovans, because the trees are linked closer or further away from the origin, that these are the SEMA, stuff from Spain, and Denisovans are more closely related than either is to the Neanderthals, and this puts Neanderthals and modern humans more closely related based on that short sequences of their mitochondrial DNA. And this becomes then gymnastic, trying to figure out how migration and human biology figured that, or led to this situation. This is one of the cases we could have, just one of these, sort of a phylogram. Look up here. We have the ancient condition with modern Homo sapiens. At some point, the Neanderthals and the Denisovans split off. This would be more, even though it's the mitochondrial model listed here, in fact, this would be more like the nuclear DNA. But then we find that this perhaps was a separate tree, that these guys went into Eurasia, and then the modern Homo sapiens came in later. So again, we could switch these connection points all over the place depending on the type of data that we're using. The other point that we're finding here is that we need more data, and we have to explain this biologically. And then things get interesting. So we have three populations. Here's modern humans, the Denisovans, and then we get the Neanderthals here. And each one of these colored zones represents the samples from the Neanderthals, the Denisovans, and modern humans, and links them together based on how close they are in terms of their DNA sequences. And what we find here is that the Denisovans are similar to individuals from Asia and Oceania, but not so much to the Europeans. What this means is that individuals from Oceania, which is Indonesia, Melanesia, Australia and such, they share 3.3 to 6% of their common DNA. If we look at Neanderthals and modern humans, non-African humans, we see that we share anywhere between 1.5 to, some estimates are as high as 4% of the DNA. And so the question is how do we share this level of DNA? Where did this come from? Well, this is that Veni Vidi Copuli, is that what we find is that the Denisovans and the Neanderthals were living in Eurasia prior to 60,000. Then when modern Homo sapiens came into this area, what they did was the populations met up and they interbred. And so that these levels of genic similarity are a consequence of interbreeding between these different populations. And so that the paleontologists have always been concerned whether they can identify inbreeding in Neanderthals. There are a couple of fossil specimens from Spain and France that look like they're part of their morphology is a little bit Neanderthal and a little bit Homo sapiens. And is this anatomical evidence of interbreeding? Well, now what we can find is that in fact we have in all of our genes evidence of interbreeding between these populations. And what we find is that with the Neanderthals, again, the Neanderthal genes are in the non-African populations, because what we find is after the humans left Africa, it's only then is when the inbreeding occurred. So we're not finding these Neanderthal genes passing back into Africa. Similarly with the Denisovan genes, and the Denisovan genes are also not found in Africans, people of African or recent African ancestry, simply because what we're finding is that this interbreeding occurred after modern Homo sapiens made it out into Africa. And in fact, you can go online and buy tests to figure out how Neanderthal you are, and I'm sure soon enough you'll be able to find out tests how Denisovan you are as well. And I don't think it's a bad thing. And so the other things we can do, and then we can actually identify what the genes are and what they code for between the different species. And I think that then we can look at what anatomies are changing over time, or what physiological processes, or what metabolic processes are changing over time by identifying the differences and the similarities between these different populations. And so what I'd like to do is, that's the end. Thank you very much